on the list.
Okay, perfect.
We are actually two different projects, so you start.
Yeah, so I mean it's kind of the same. We found out it's we're both trying to do gene expression.
Okay, and I know that Brooke, did she reach out to you, Mara?
Yes, so I have so far Valentina, Brooke, and I
Okay.
have other two hopefuls that want to come in, but not yet. They don't know now yet.
Okay.
They don't know about the project yet. So.
Okay.
And my group also has two other people. So
Who's
it's Taylor and Bodo.
Okay.
But I wasn't sure if you were allowing a group, like a combined group, or if it's
I think
similar.
if you guys wanted to do something very similar, that is fine.
Yeah.
I definitely don't want more than like 4 and 5 people in each group, right?
Oh.
So like, let's say you guys are both doing cloning. You can do very similar or the same projects, and you're both doing it, right? Each
Okay.
level is doing it. That is perfectly fine to me. We all know how groups work.
Yeah.
I don't want one person doing the work for 10 people,
Yeah.
right? I want you guys to actually learn something.
I was thinking I could be the supervisor. No, just kidding.
Cool.
Now, what I was thinking actually was like we could do like the same, but then they could do one hypothesis and we do another one. You know, like.
Okay.
But let's see. Why don't you go
Yeah.
with your idea, because maybe you have your own idea and it's great.
Yeah. It's it's gene expression too, but we were trying to see if we can do gene expression of a striped strand from E. coli, maybe something like yeast or SARS-CoV, and try and see if we could not the whole pathogen, but a strand of it, and express it, and then eventually purify it. I think I overheard that purification might be something a little too ambitious for the time span that we have.
It is possible. So purification of a gene expression, you have to learn how to do gels. It's not horrible. Okay. If you guys could purify it and do a gel, I think that is probably about what you're going to get to as an experiment.
Okay.
Nothing carcinogenic or biogenic. So no COVID.
No COVID. Oh, okay. I was,
the last thing I need you to do is create a new mutant strand of COVID.
The EBCC lab.
Originated number.
We already
and
had
then
it
another one.
COVID leave Everett once before. We might as well do it again.
Okay.
Now using yeast and doing an isolation and gene expression, beautiful. So we can do that really easily. You can design a plasmid. Whatever you're trying to do, we can do that. That is pretty reasonable.
Okay, perfect. Because I was thinking we could do it in vitro or doing some in silico, like doing some kind of model, like a online model too. But
yeah.
That was kind of like a second option.
So I think I doing isolation and yeast sounds wonderful.
Okay, awesome. I'll get with my group. We'll talk about it.
Give me one half of a second. I'm gonna give you some information. There's a lab that we've been thinking about building. Okay. Where's the chat? They change this Zoom on me all the time. There you go.
Oh, perfect.
All right, so there are, honestly, and this is one of those that I think you should learn to use. Ask ChatGPT how to do it.
Okay.
Okay? ChatGPT will tell you the step-by-step instructions. Now, you're going to have to go and learn a bunch of things. You're going to have to add to it, but it will help you out.
Gotcha. Yeah, we'll I'll send this to my group and see what they think about it. But
Sounds good.
I feel like they'd be open to do yeast too.
Yeah, I think it would be fun.
All right. Awesome.
And could do two different things, right? So there could be two groups doing something very similar. And it'd be reasonable.
Right. Is it possible to? I know. I think the science fairs I've seen so far, they've been the trifold ones. Can we also do that and like a little like model of some sort, like kind of like?
You could have a model. We do for biology. We print the posters.
Right. Absolutely.
Look at what's on the wall.
Gotcha.
Yeah.
Oh, perfect.
Okay.
So you're gonna do exactly what's on the wall outside the lab.
Got it. Okay. Gotcha.
So is that it?
Yeah, that's it from me.
Okay.
I'd have to do more research about yeast than what he just sent out.
Yeah. Okay, so for our group, what we wanted to combine was cloning, PCR, and art.
Uh-huh.
Okay, so we have 4 hypotheses. The first one that I'm going to tell you, I think it's been done already. So it says clone GFP, which is the green fluorescent protein gene, into E. coli and induce protein expression. The bacteria will fluoresce under UV light, allowing to visualize gene expression. I think that's been done. So.
Yeah. Everything you guys are doing has been done a million times, right? Mm-hmm. So you're not inventing anything, you're not doing it, but you're learning a new skill set. So perfectly reasonable.
Okay. And the other one is, the other hypothesis is different, which is the one that my group is leaning towards, is different temperatures affect GFP expressions in E. coli. Hi, Sylvie.
So that would be different than, so if you just grow GFP and make art with it, okay, that's a really simple experiment. And you can illustrate your art. It's a lot of skills. It's gonna take a lot of time to learn, okay? Mm-hmm. Which is very doable. If you're wanting to do GFP at different temperatures, now you not only have to learn all of those skills that you're gonna do for the art, but now you have to learn how to manipulate at different temperatures, right? And isolations. So that might be a little bit more advanced than what you're wanting to do.
Oh, the temperatures?
The temperatures.
Yeah, because we were thinking about, combining it with climate change, to see how.
Yeah, but
now I know ChatGPT doesn't know it all, but it said it was doable for 6 weeks, but no, and for our levels.
I don't think so.
You don't think so?
And then you'd have to ask yourself, why do we care about growing yeast at different temperatures? Climate change is never going to affect yeast. Yeast is grown in different conditions. So, and do you know anything about where the glow fluorescent protein GFP was developed?
From either plants or bacteria, no?
UW, Fire Department Labs discovered it.
Oh.
So it's a marine thing.
From where?
It's a marine thing.
Oh, okay.
And so there's so much known about how it fluoresces and all of those things already. So I don't think your guys' project is learning a skill, right? That's your guys' project. So probably more the first one is more doable.
Okay, let me tell you because I have 2 more theories. Co-expression of chaperone protein improves proper folding and brightness of GFP into E. coli.
Sounds wonderful. Sounds like a graduate project.
Sounds like a what?
A graduate
project.
Oh, no. And there's another one. Non-sugar inducers, sugar substitutes like xylitol or stevia can trigger GFP expression via ADA-BAD promoter.
Again, it sounds neat. So you're just going to grow those yeast with under different conditions, see if you have GFP produced. That's simple in itself. Now, are you gonna have to clone it to do a plasmid to insert everything for that promoter? Or are you finding a strain that already has that?
We're researching about it, so.
Okay, because if you have to design it and insert it, I would say that's probably gonna take 6 months.
Oh, yeah, okay. But but it's doable if we buy it.
Possibly. I don't know if there's any strands out there that have that specific thing.
Okay. It says via ARA bad promoter. I don't even know what that
I
is.
don't know what that is.
Okay, maybe that's one of the Astras. I don't know. Anyway, and it was combining it with art, vary sugar concentration, temperature across plate,
Okay.
map going zones into patterns. Science meets art.
Yeah, see, growing things at different temp and induction for plating, that's what I was thinking you guys are doing.
Can you repeat that, please?
So exactly what you're just saying, it's the art, by plating things out, you can draw things, you can induce with temperatures, things like that. That's
So-
how you make an art project with it.
Oh, okay. So in this case, we do use temperatures, but it's just more like to get colors or
Yeah,
types?
get colors, get induction of the fluorescent proteins.
Okay. So you're saying the first hypothesis and then combine it with art.
Possibly, yeah.
Okay.
And what's really kind of what you're going to do is you're going to insert a plasmid, right? And you want this to grow under a certain condition, and you are going to draw it on a plate. And if your art project shows up on the plate, your experiment worked.
Wonderful. I love that.
It's fun. It's neat. You learn some experiments. You learn how to do plasmids, things like that.
Do we learn to
Very-
clone?
Yeah, that's what cloning is. Yes.
Okay. And it's got PCR and everything.
Yep.
Okay, wonderful. Okay, my group, I actually went a little bit ahead of my group that they're gonna probably wanna meet again at 12:30 if, I don't know.
Okay.
I'll tell them, but yeah, they'll probably have some
Okay.
questions. Thank you.
Okay.
Thank you.
All right.
All right. Next one on my list is Kenneth.